We adhere to the highest standards of professionalism and ethical practices. I, Dr. Indu, on behalf of the entire team of School of Virtual Forensics, wish to thank all of you for making time out of your schedule to join us for this webinar. And I would also like to thank our team of members, guides, and mentors without whom the whole enterprise taken out. The topic we're discussing today is forensic genetics and its legal importance. We will deliberate on this topic, which will be taken up by Dr. Bhargav Patel, sir, who is our speaker for the day. A little bit about Sir before we start. Sir is an assistant professor at the Institute of Forensic Sciences at Gujarat Forensic Sciences University, Gandhinagar, Gujarat. Sir has completed and done extensive research in the field of biotechnology, microbiology, and molecular biology, and Sir is an excellent academician with over 9 years of teaching experience, in addition to being a researcher. Sir is also a guide and mentor to PhD students for the past 3 years. Sir has also published over 10 research articles, 3 chapters, and also has a patent to his name. Along with that, we have a Sir who is a member of various scientific communities such as the Biotech Research Society of India, the Indian Science Congress, and the Indian Society for Human Genetics. Again, this is just little of his achievements to name, but we don't have enough time to elaborate on Sir too much because he has done a lot of great things for the field. So we'll let him take over and we'll commence with his lecture. Welcome, sir. Thank you so much for joining us. So if you would just take over from here.
Yeah, thank you, Dr. Indu, for such a nice introduction and very kind words for me. So I hope I'm visible to everybody and audible to everybody.
Yes, sir.
Okay. So before I start my presentations, I'm really thankful to Virtual School of Forensics for inviting me for this lecture and considering me an expert in this field. Though I am very new in this field, but because of the teaching experience, whatever the knowledge I have gained throughout my this career, so I would like to share some part of that particular topic today, and that is forensic genetics and its legal importance. So let us start with that topic straightforward without taking much time. So I'll share my PPT with this presentation. So let me share this PPT with all of you. Just a minute. I hope you can see my PPT.
Yes, sir.
Okay, so everybody is okay. So forensic genetics and its legal importance. See, forensic science is a very dynamic field and it's a multidisciplinary field. So it has lots of application of biology for investigation purposes. And genetics is not you can say it is not an exception. So and it is, you can say, one of the most important aspect of the investigation nowadays. Why? Because of the DNA, right? So let us see how this forensic genetics flourished and how it has helped the justice delivery systems in a very great way. So let us start with the, just a minute. So these are the contents of my today's talk. Introduction, then I'll be elaborating what is exactly forensic genetics and what is the basis of individualization. The third topic is DNA databases and its legal aspect. What are the contemporary development? What are the modern equipment, techniques, and modern aspects which are going on in forensic genetics right now, that we'll be discussing. And finally, we'll discuss about the limitations and challenges which are there in this particular field. So, uh, if you see, the curiosity is the driving force of knowledge. So that is evidenced by lots of scientists and lots of visionary. We have given a lots of knowledge because of their curiosity to know the world, to explore the world, and to know the facts of natural principles. Right? If you consider Acharya Kanad, if you consider Aryabhatta, or you consider Darwin, all of are the curious people which gave us a very good wisdom and in-depth knowledge about the scientific principles which are there inside the nature, right? Then, if you go further, so Mandel, Frederick Miescher, T.H. Morgan, and finally Watson and Crick. So if you see the timeline, so in 1900, the rediscovery of Mandel was the initiation of the era of genetics, right? Actually, the work was done in 2020, right? But the work was rediscovered by three independent scientists, and because of that, we came to know about the genetic principles. Then in 1869, Frederick Miescher isolated the DNA. You can say the crude mixture of the first cells from the first cells, and that he called nuclein. Then in 1911, T.H. Morgan derived that the chromosomes are there, which are responsible for development of flies and all that. And finally, in 1953, the structure of DNA was invented by Watson and Crick. See, during this particular timeline, lots of other events also occurred, but I have just selected few one. So, just to give you an idea that how much efforts were given to develop this particular field. Then finally, come to that event that gave the birth of modern era of DNA fingerprinting, or you can say the modern era of forensic genetics. If you means everybody is remembering this guy, right? Mr. Alex Jeffries, who invented this particular technique, DNA fingerprinting, right? So if you see the first publication he did in 1985 in Nature, individual specific fingerprints of human DNA. So at that time, fingerprints were only the entity which were unique into the human being. So that's why he called this DNA pattern fingerprint, right? And in 1986, he took up the first criminal case, and which was convicted in 1986, and that case was of Colin Pitchfork case. This is a very well-known case. What happened in 1983 and 1985? Two young ladies murdered, and both the semen samples, both the modus operandi of both the cases were same, and from both the sides, the similar semen samples were derived. When I'm telling similar semen samples, so at that time in terms of blood grouping, at that time DNA fingerprinting was not that much developed. But finally, they took the help of Alex Jeffries and finally solved this case. Before this case, there was one case that was an immigration case in 1985. Actually, that was the first case which was taken up by Alex Jeffries to solve that immigration case. So the first was the paternity case, right? But the first criminal case was in 1986. And if you are more curious, then you can go to this movie, Code of a Killer. I think probably it is available on Amazon Prime. So it is completely based on this particular case, 1986 case. And it will give you the very good idea that how DNA fingerprinting technique was developed and finally it worked, right, in 1980s. Now, let us discuss about the forensic genetics in somewhat detail. So the definition, if you see the forensic genetics, the application of genetics to human and non-human materials. It is in sense of a science with the purpose of studying inherited characteristics for the analysis of inter and intraspecific variations in populations for the resolution of legal conflicts. So this is the complete definition of the forensic genetics. So basically, it is nothing but the application of genetics to solve the cases which involve some biological material from which we can characterize some inherited characteristics, and finally we can match it, or we can corroborate it with each other, and we can get some idea about that. What are the applications? See, if you want to know that particular science, first of all, let us see the applications. Then we can go for the principles. If you see, so ranging from the individual identification, you can see the human remains. So how to identify these human remains? Anthropological data are there, but if you are getting only piece of bone, how will you identify that? So forensic genetics is helping that. Right, mitochondrial DNA analysis, so that maternal inheritance, that you can get it done by forensic genetics. DNA barcoding, that is one of the you can say most important applications in terms of diversity studies and non-human DNA analysis. If you will say the paternity analysis, so that is another application for DNA fingerprinting and forensic genetics. And finally, you can see the microbes are also not an exception. The microbes are also helping legal aspects, and you can say in legal area. So that we will discuss later on. But these are the applications. But how these applications are possible, right? So there are lots of questions to all of you, that what is the basis of this particular technology? Using what they are doing this technology? Which softwares are used? Which mobile apps are used? You know, many young groups are there. I'm also young, right? Many mobile apps are, so they must be thinking, oh, so is there any mobile app by this we can do the genetic analysis? Yes. Then which techniques are utilized? So all these questions, let me answer those questions one step by step. Okay. So these are the prerequisites I would like to have with you, right? At least you should have an idea what exactly meiosis is. You should have an idea what crossing over and recombination is, and what are the basic Mendel's laws of inheritance. I'm not going much more detail, but you must be knowing that meiosis takes place in the reproductive cell, and because of the meiosis only, we are having the haploid cells. You can say the daughter cells here. And during this meiosis, a wonderful mechanism is occurring, which is which gives rise to a diversity, and that mechanism is known as crossing over and recombination. That is the only mechanism, in that particular process, which is responsible for the diversity on the earth. For I'm talking about the human. I'm talking about animals. Means most of the animals, right? Those are eukaryotic groups. This particular mechanism is occurring there, and this is the basis of our you can say DNA fingerprinting technology. We'll see later in much more detail. And the Mendel's laws of inheritance. So that independent assortment and some other laws, right, which are there. So if you're having this background, then you will enjoy this lecture more. Let us see the basis of individualization. So everybody is knowing that DNA fingerprinting is utilized to identify a particular person, a particular human, right? So that individualization is there. Then how this individualization is taking place, right? So that is very important questions to be answered. We are all unique. You know why? Why we are all unique? Can anybody say? Anybody? Turn on your mic and you can say. Anybody? Why we are all unique? In even twins also. So what exactly the basis of this particular uniqueness? Can anybody say?
The genes difference.
Yes, genes difference. Yes, obviously, right.
Seen among the people with different races, origins.
Very good. Yes, yes. So these genetic variations, right? So ultimately, whatever the phenotypes are there, we all know the central dogma of life. DNA gives rise to RNA. RNA is converted to the protein, and this protein is converted to the structures, and finally we are made up of these proteins, and this is how our shape is derived, right? The nose shape, the eyes color, hair color, beard, lot many characteristics on my body, inside my body, is because of the DNA, right? So these variations are important, and these variations are there. And how these variations are occurring, there are lots of mechanisms, and one of the most important mechanism I already stated, that is meiosis, that is crossing over, that is recombination. Okay, let's go further. So whatever the genes are there, so this genes undergoes variations. So say for example, if I am having a particular gene, that you are also having a particular gene, but that may be variable. I'll give you an example, a very good example that I think everybody must be aware of, tech DNA polymerase, right? So that is DNA polymerase from Thermus aquaticus, which is thermostable. And what about the human DNA polymerase? The work of both the gene, work of both the protein is same, DNA polymerization. But one is thermostable, but another one is not thermostable. So this kind of variations is there, right? So that genes leads to the variation, and this variation leads to the allele. So what does it mean? There is an allele, one allele which is there for thermostability, and another allele which is there for the thermo instability. So that particular whole mechanism is known as polymorphism, right? If you still not clear, then this is polymorphism. All are minions, but they are different, right? All are minions, but they are different. Here also, if you can see, they are shells, but they are different. So how this variation occurring? Because of the polymorphism, right? So now another question: what kind of polymorphisms are there in our genome? Anybody? What different types of polymorphisms are there? Okay, let's go further. There are three different kinds of polymorphisms which are there, and because of that variations, we are different. The first is, you can say, size or length polymorphism. Everybody says that 99.9% genome of every individual is similar, right? We know that our genome is containing 3 billion base pairs. Out of this 3 billion base pairs, 99.99% is similar to everybody, is identical to everybody. But there is a 0.1 variation is there, and that variation is because of these three types of variations. You can see the size poly, whatever the DNA sequence is there, it may have some repeats, and because of this repeats, right, it may have 3 repeats, and one DNA sequence have 2 repeats. For example, I am having 3 repeats, and another person is having 2 repeats. So there will be size difference. Here you can say 9 base pair, but here I am having only 8 base pair. Oh, sorry, here 12 base pair, and I am having 8 base pairs. So that is called a size polymorphism. That the application of this size polymorphism is described by VNTR, STR, and other repeat regions. VNTR is variable number of tandem repeats, which were used by Alex Jeffrey. STR, short tandem repeats. So after the Alex Jeffrey to deal with the VNTR, so that's why the STRs were invented, right? That are short tandem repeats. We'll take a look over STRs in somewhat detail. Then other repeat regions, we know that out of this 100% genome, 3 billion base pairs, almost 70% sequences are repetitive sequences. So we are talking about these repeat regions, and that also lead to the size polymorphism. Another polymorphism is sequence polymorphism, right? The size is same. For example, once again I take the same example, DNA polymerase, size is same, right? But there is a difference of few bases, right? You can see here C is replaced by T, A is replaced by Z. Then the codon will be different. We know that each and every codon is transcribed or translated into the amino acid. If one codon is changed, amino acid gets changed, and finally the protein structure will be changed. So this kind of polymorphism is there, and if the protein structure is changed, you are having a variation, right? And finally, the copy number variations. So multiple copies of the gene. We know that few genes in our genomes are having number of other means multiple copies. For example, housekeeping genes. Those housekeeping genes are having multiple copies, and we differ in that multiple copies also, thanks to recombination, right? And because of that copy number variations, we differ in metabolism also, right? I may metabolize milk very slowly, but somebody other may metabolize milk very fast. Why? Because the casein hydrolysis or some other enzymes which are responsible for the milk metabolism are more in that genome, but it is less in my genome. So this is called a copy number variations. So we will have a very good example when we'll discuss about the molecular autopsy. So these are the three different variations are there, and if you consider the sequence polymorphism, there are three major variations are there. That is called SNP, single nucleotide polymorphism. So in a complete gene, there is only one base is changed. A very good example for that is the sickle cell anemia. Right, it's a very wonderful example. That only one codon is changed, and completely the shape of red blood cell is changed because of the one amino acid. So that is called SNPs. And indels are there, insertions and deletions. So some because of this particular insertions and deletions, the size may remain same. For example, this is called how this G is placed. So A is deleted and G is inserted. So these this is how these insertions and deletions are there. So these are the three variations which are responsible for the complete diversity of the human. So these is making the basis of forensic genetics. Just say the hello to the DNA structures. Everybody is knowing about the DNA structures. I'm not going in much more detail. You know that the nuclear DNA remains here in the in the nucleus. Mitochondrial DNA remains in the mitochondria. We are having two types of DNA: mitochondrial DNA and nuclear DNA. The DNA is a double helical structure, and which is having four basic alphabets A, T, G, C. These are nothing but the nucleotides which are joined together by hydrogen bonds, and they are joined in a chain by phosphodiester bond and making a you can say nuclear chain, and they are connected with the hydrogen bond between. So A with T and G with C, triple bond G and C, double bond A and T. So we know about that. So this DNA is the main basis of forensic, this individualization. Let's go further. So this is somewhat more about human genome. So you already know about the 23 pairs or 46 chromosomes are there. Total 3.08 billion base pairs are there in our genome. These are the different chromosomes, and they are in pair, and that's why 46 chromosomes are there. Chromosome number 1, 2, 3, 4, 5, like that. And you are having this reproductive cells, and here exactly in the reproductive cells will decide the sex of the human, right? Whether male or female, right? And they are differentiated based on the size of the DNA material that they are containing. Now, if you combine the complete all the chromosomes, and if you make a long string of that, it will be 6 feet long. So you can imagine that this 6 feet long string is highly coiled, and it is remaining in the nucleus. Okay, so this was about the human genome. Now about the short tandem repeats, and which are the main markers which are utilized to identify a human. How it is basically identified by STR. So you can see here 3 different people are indicated here. One person which was having a short tandem. Short tandem repeats are nothing but you can say this here AGAT. These are the tetranucleotide repeats, right? They are repeating continuously 1, 2, 3, 4, 5, 6, 7 times. Okay, so this particular piece of DNA is having 7 repeats on one chromosome, but this gentleman's another chromosome, the AGAT is repeated only 6 times. What does it mean? It means in one chromosome it was given by his father, and on another chromosome it was given by his mother. So this particular 7 repeats and 6 repeats is making the genotype for this particular marker for this gentleman. Same thing if you consider the same marker AGAT continuously 4 times repeating, 19 times repeats. So both these persons are having difference in the size of DNA at this particular loci by, you can see here, 4 repeats. It means 4 multiplied by 4. That is called 16. So 16 base pair. Here it is 19 multiplied by 4, that much base pair, and here 16 multiplied MF4. So this is how the size differences are there, and which can be detected on the electrophoresis. And based on that, you can differentiate this particular two persons. That okay, this is the profile of this person, and this is the profile of this person. So both the persons are not matching with the profile, and that's why they are different. Similarly, here third person, if you'll see, so 12 repeats, 14 repeats. So now there must be a question that how many variations are there in this particular marker. So it depends on the marker. For example, this AGAT having, for example, 40 variations. What does it mean? 40 repeats, right? It is going minimum 7 and maximum 40. So 40 times. So if you minus it, 40 minus 7, what will happen? 33 different alleles are there. Similarly, for here, so out of 33, any two alleles identification of the human, right? I know that you are many of you are still not clear how it is being utilized. No problem, it will come further, don't worry. Now this is very you can say good example that which are the matching criteria. For example, here the matching criteria was only one marker. So based on one marker, for example, take an example of blood group. So how many persons you can identify in a group, right? On the basis of blood group. So there are 9 or 10 blood groups which are available. So if more than 10 people are there, then what will happen? The blood group will be repeated. And that's why you cannot distinguish the person if the more than 10 crowd is there. So what does it mean? The matching probability, or you can say the maximum matching probability of that particular marker is 1 in 10, for example, based on blood group. But if you are having 33 alleles, so what you can do? Homozygosity and heterozygosity. If you can calculate, then it will be more than that, right? But that is not enough for us. Why? Because we want to identify one person in total population of the whole world, right? So total population of the whole world is 7 billion, right? So how can you distinguish one person in 7 billion, right? So you have to depend on some other criteria also. So what different criteria or what different markers can be utilized? So I'll give you example. Right. For example, if you are searching Amazon and you want an Android phone, right? And if you will type an Android phone I want, right, the result will be like this. You will be given lots of options, right? Right. There are thousands of models which are available, which are running on Android application. But if you will increase another parameter, I want 4 GB RAM, right? It will cut down this. So out of those whole junk, now you are having these are the models which are having Android application also and 4 GB RAM also. Now, if you add the third parameter, I want 128 GB, not RAM, sorry, here I have misspelled, not RAM, but the storing capacity, right? If you are matching it, then still now you are having only 6 models which are having these 3 criteria. Now, if you are at 2 more parameters, then you will come to know the individual mobile set that you are looking for. For example, I have added 48 megapixel camera and the company name Simpson. So if you are having this 5 parameter, or you can say the 5 marker, then you can identify this particular mobile. I hope you are clear now. So if you are having this 5 marker, right, persons, right, for example, if you want to identify a person with hat, with goggles, red shirt, white shoes, and black pants, right? These are the 5 criteria. Then you can identify this person in a crowd. So this is how the marker works, right? So the probability, if you are just looking after the person with the red shirt, so in the world, many people are wearing the red shirt. But if you are adding more criteria, there's a red shirt but hat, then you can narrow down. If you add one more, narrow down. Just like your another example, just like your address, right? City name, society means street name, society name, block number, and finally your flat number, right? So that will be individualizing your home. Similarly, we are going for human also. using these different markers. These are called core STR markers, right? And if you will see, each and every chromosome is having one or two markers, right? If you'll see, these are, if you see here, two colors are there. One is yellow color and one is orange color. So initially there were 13 Codis core loci. Now, what is Codis? Codis is nothing but the database of USA. DNA database of USA, combined DNA index system. Okay, so we'll discuss later on. Now, up to 2017, there were 13 Codis core loci. It was decided that okay, when you want to analyze any human samples, then minimum 13 marker must be matched in order to admit that particular report into the court, right? But now from 2017 onwards, it is revised up to 20. So these are the 20 markers, and which are the orange one, there are latest addition into this marker. So whenever you want to use this identification technique, then minimum 20 markers you must use in order to get the identity. Why? To not make an error in identification of a person. Why? Because absolute identification is not possible at all. You cannot tell that is a 100% match, right? Many of you are maybe unaware that whenever the DNA report is submitted into the court, it is never mentioned that there is a profile match. It is never written that is a 100% match. So, okay, the DNA profile of this person is matching with this person. No, it will be always mentioned in random match probability. That I'll discuss random match probability also, how to derive that random match probability and how to use it in a you can say jurisdictional purposes. Okay, so I hope everybody have understood this use of multiple markers, right? And these particular markers are mixed together. The primers for these markers are mixed together and they are utilized for amplification of the DNA. So these are the markers. So this is the basis of the individualization of the person. Now the techniques. I will not go much more detail about the techniques, right? A little bit about the techniques. You can say first, so these are the tools of the trade and flow of the process. First of all, collection, preservation and transport of the crime scene samples from the crime scene or any subject or any person. Then the DNA will be extracted. Then the amplification of the DNA will be done by PCR method. And finally, the STR profiling will be done on genetic analyzer, that is called capillary electrophoresis. And finally, the interpretation of that particular profile. And then preparing a chart showing the marker alleles and finally matching it with each other, right? And then find out the random match probability and finally preparation of the report. So let us see one by one. So that these are the collection tools. So these are the swabs by which you can collect the any bodily fluids from the crime scene, which is dried or wet, right? Using this particular swabs. Varieties of swabs are available, which is having lots of advantages, that antibacterial material, antimicrobial material, which is having a perforated, you can say, to cut it down, and finally dry it and you can send it. Cotton swabs are having somewhat difficulties in releasing the material, but you can say nylon material which was utilized in a modern swabs, which can give you the good DNA. Then these are another varieties of, you can see, the self-drying. There is no need to do the drying. You can just put it directly inside and send it to the laboratory. There is no need to do drying, because during drying process also there are chances of contamination. And one of the best technique to preserve the DNA, collect the DNA and extract the DNA. 3-in-1, that is FDA card. So I think everybody is knowing about the FDA card, so I'm not going much more detail. They are impregnated with some chemicals, right? Some cocktails which are destroying the cells and finally entrapping the DNA into their matrix, and finally whenever it is required, it is releasing into the PCR mixture. So this is how this FDA card, and you can store it at a room temperature. Then storage and transport. So you have to take care that the moisture should not be there, because it is one of the most important precautions you have to take, because whenever the water activity will be there, bacterial contamination will be there, and finally your material will get destroyed. So that's why, because it is a biological material and it is a source of source of nutrition for the bacteria, and after the gut, bacteria are everywhere, right? They are the second entity which are everywhere. Okay, so that's why we have to be very careful from that bacteria and microbes, other microbes. These are the extraction kits. Modern kits are available from many distributors. Right, I have just mentioned three, which are the major players: Thermo Fisher, Promega, and QIAGEN. So any kit you are choosing, which is working for different samples and different category of the samples. From, you can say, direct extraction can be also done, and processing also there. Silica gel based technologies are there. These are the modern instruments which are doing direct extraction, automatic extraction. Just you need to lyse the cells and finally put it inside that. The purification steps will be taken care of. So a few machines are also there which are doing lysis also and finally purifying the DNA also. And which will be, these are the basically utilized in high throughput laboratories. Yes, the genie of molecular biology, PCR, which is one of the most revolutionizing technique which has affected the forensic genetics a lot. Why? Because without this PCR, most of the DNA samples at crime scene will not be possible to analyze by forensic genetics. Why? Because a very low quantity of the samples is the problem, right? Whatever the Alex Jeffrey created or invented the technique, which was dependent on the high quality and high amount of the DNA, RFLP, right? So that technique is not, it is utilized for some other applications, but not for the DNA fingerprinting nowadays. The PCR based techniques are utilized, right? So what we are doing is, we are not analyzing the whole human genome for identification purpose, but out of that whole human genome, we are identifying only 24 markers. Nowadays, most of the kits are having 24 markers. So we are amplifying those tiny regions, 24 regions only. It is, it's 0.00001% of the total genome size, and that is being amplified, and that characteristics is making our ourselves, the humans individual. Then these are the STR amplification kits. Ready-made kits are available because it is very difficult to manage the multiplex reactions. So that's why ready-made kits are available. You can see all the markers are there. 24 markers are available, and in this particular size of the block is showing the range of the alleles from particular base pair to particular base pair, right? And these colors are showing the different color channels. So they are labeled with this blue, green, yellow, red, and once again light green colored dyes and fluorescent tags, and which can be detected in the instruments which are there. This kit is from Promega. It is also having 24 markers, right? And which is having once again the 6 channel, right? And multiplex kit. Investigator 24-plex kit. This is from QIAGEN. So these are the 3 major players which are having this particular kits available with them. Now, finally, after the PCR, DNA is extracted, PCR is done, polymer is. So how to detect which marker is amplified, which allele is present in that particular person? So this instrument, capillary gel electrophoresis, known as genetic analyzer also. Basically, the capillary will be separating the DNA marker based on their sizes, and that will be color separated. And finally, this is the raw data, and the pics will be detected. And finally, this pics will be converted from time scale to size scale, then from size to allele, and finally the report will be generated by software. So which kind of report will be generated? This kind of report will be generated. So that is known as either genotype or profile or DNA fingerprint, whatever you tell. This is the particular thing which is known as genotype, profile, or a DNA fingerprint. Right. Basically, this is the raw data, means the analyzed data. You can see here, this is the blue channel, this is the green channel, this is the orange channel. So different channels are there. This is just half of the report. You can see each and every marker. You can see here one allele is there, here 2 alleles are there, here 2 alleles are there, heterozygous. You can see here the heterozygous samples are almost equal with each other, and whenever there is a homozygous, it is double the size. You can see the size, of course, because on both the chromosome amplification has occurred, and that's why this product is having double the concentration as compared to this product, because it is divided into 50-50%. Okay. And finally, this particular, on the basis of this report, this particular report will be generated. You can see here, this 24 markers are there, which is listed here, and this is the sample name. I've given the one analysis from our lab. So you can see these are the different markers names, and these are the alleles which are present. 15 means, here it is 0 means no allele. So here it is homozygous, heterozygous, heterozygous, like that. And when it is X, so here one marker is there that is called amylogenic, which is sex differentiating marker. So you can see here X and Y. Y is missing, so definitely it is a female profile. And finally, these are the different allele sizes. So this is the DNA fingerprint. Now, another sample you will take from the crime scene or a reference, and you will match with one. But this is not actually the process. I'll show you the process in detail. Now, let us see about the DNA databases and legal aspect of this particular profile. So now, this profile can be generated from crime scene samples, from reference samples. I hope you are clear when I'm telling reference samples. What is reference samples? Either suspects are there, or if it is a paternity case, then the mother samples and father samples and that son or daughter samples. So these are known as the reference samples. It means healthy persons from the samples which is taken from the healthy persons, or means the persons which was victim or perpetrator, right? And crime scene samples. So the profiles are generated, but how it is having a legal importance, right? So I hope now you are clear that in jurisdiction, in the courtroom, which is acceptable, that whenever you are having a uniqueness, when you are showing that this particular material is directly connected with this person, you are linking that person with the crime scene. At that time, it is admissible. So DNA is having that uniqueness, just like a fingerprint, and that's why DNA evidence is admissible in the court. Now, DNA is admissible in the court. So now there was a requirement. Actually, UK was the pioneer. So this technique, Alex Jaffe was from UK, and the DNA fingerprinting started in the UK. So UK was the pioneer in this particular technique, and Britain was the first country to implement the DNA database. What do you mean by DNA database? So what exactly they do is, whatever the DNA samples which are collected from the crime scene, from the perpetrator or from the victims, will be that which particular profile should be stored in the database. It means it will be stored in the database, and whenever you want to search against that database, you can search it. Just like we are searching, Control F. We are searching Control F, and you are putting some word, and in that file, whenever it is a match, it will show the match. Right. Similarly, DNA databases are also made. Right. Let us see the legal importance of this DNA database. First of all, how to match, that I was talking about. Right. So this is your SCR locus. I have just shown only few markers. This is the evidence sample which was collected from the crime scene. Now suspect A and suspect B are there. Now you are matching with 1517. You can see here it is not matching with this suspect A, but it is matching with suspect B. Similarly, 1516, it is not matching with this, it is matching with. So if you see, the complete profile of evidence is matching completely with the suspect B. Now, here the final column you can see here. This is the final column, right? Which is very much important to understand. Why? Because you cannot say directly that it is matching. The reason is, still it may be a random match probability. It may be possible that randomly this evidence sample was matched with this particular suspect B because of some, you can say, we know that because of the recombination and because of the crossing over, it is impossible to be matched between two persons. Why? We know that diversity, the concept, but still we cannot tell. It may be whatever the probability is there, but probability is there that randomly two samples can be matched. So that's why in the court, when we are looking at to that, so we have to, accurately present that particular profile. So how to that? So you can see here the suspect's B's genotype frequency. Now, what do you mean by frequency? That 15 and 17, right? So that is a genotype. It means how many persons in the population which is having 15 and 17 allele at this marker. So the probability of this particular in that population is 0.13%. What do you mean by that? That out of the total population, 0.13% people are having the same profile, right? So based on these marker, you cannot tell that evidence sample is completely matching with the suspect B. Now take another marker, right, 15, 16. Now it is having 0.2%. So what is the probability if you combine this to 0.13 multiplied by 0.22? Why? Because both the alleles are independent with each other. So another rule, independent assortment, and that's why the multiplication rule will be applied here. So you can see here 0.13 multiplied by 0.22. So that will make, you can say, the much more broader, you can say. So, but still it is not, you can say, convincing. So take third marker, combine the frequency. Fourth marker, combine the frequency. And finally, when you combine the frequency, if you multiply by this frequency with this, then this, then this, then this, you will have a very big number. You can say it may be, 0.1.
Sir, not audible.
Sorry. Hello.
So please continue. You're audible, sir. Yes, sir. Kevin, sir, could you please check the system settings? Maybe you're having some disturbance.
Okay, no
so
problem.
Please continue.
Okay. So this random match probability will be calculated by multiplying each and every frequency of genotype. And finally, and how you got this genotype frequency? Based on the frequency of allele frequency, 15 number and 17 number. You combine this, you are getting this. And finally, you combine this, you are getting one number that will be 1 in 10 raised to 16 or 10 raised to 24 or 10 raised to 64, like that, right? So this is just, I have given an example. For example, if you are taking only 3 marker, right? And how many times allele observed? So this is called allele frequency, right? And that is from particular population database. So whenever the USA is applying this data, then USA is taking the population stats from all over the world or all over the USA. It may be possible. Size of the data, you can see here, 604. So based on that, the probability is 0.34 and 0.05. If you combine them, then this is the probability. And when you combine these 3 probability, you are okay. So this is how the science and law is differentiating, right? If you will see the issue truth, then the science serves the interest of itself, but serves the interest of the which is open in science. It is privileged because law cannot be applied to everywhere, right? Process, it is unbiased and systematic. It can be adversarial, defending a specific position, right? So here you are having a chance to defend, you are having a chance to prosecute. Here it is completely unbiased. Whatever it is, we have to accept. What are the goals? So advance human knowledge, eliminate false beliefs, document knowledge through publications. Which are proving at its best will be given the verdict, right? So this is the difference between science and law, and that's why the science is helping the law to go to the accuracy level. Is knowing the case of that football player, I forget the name, O.J. Simpson. That was the, you can say, the case of century in 20th century, right? Which was important, which was there, who educated everybody about the DNA, because it was a media trial, and because of the everybody came to know about the power of DNA, and that was based on chain of custody. Actually, he murdered his wife and her boyfriend, right? Everybody was knowing that he murdered, but it was not true in the court. Why? Because of the chain of custody was compromised, and that defense lawyer proved that chain of custody is compromised, and finally that DNA reports could not be admissible in the court. So that is the common challenge. So you have to maintain the chain of custody very well in order to admissibility into court. Otherwise, it will not be possible to admissible. Then qualifications of the experts, reliability of the technique and testing evidence, reliability of the analyst performing the work. So that's why you have to maintain the quality and quality assurance at your laboratory level, and that's why accreditation comes in existence. That's why the auditing of the laboratory comes in the existence and everything. Why? Because it is meant for conviction. It is meant for you can say justice delivery system, right? Contamination of evidence, mishandling of the evidence, human error during testing, bias in testing, maybe, and that's why DNA profile is always checked by 3 individual persons independently, and then when they are coming to the same conclusion, then and then the report is submitted. Lack of information regarding when and how the DNA was deposited on an item, and lack of control over the handling. That is one of the most challenging aspects nowadays, because of the each and everybody is asking for the DNA analysis. It means every investigator is asking about the DNA analysis because it is a gold standard. It is said to be a gold standard. But what are the bottlenecks? What are the challenges that I'm going to describe later on? Okay, whenever you are creating a national data, few aspects that you have to cover, right? That a commitment on the part of each state, any local or government to provide samples for the DNA database, both offender and crime scene samples. So that is compulsory. A common set of DNA markers or standard core set so that results can be compared between all samples entered into the database. So just like USA did that 20 core codis loci. Similarly, any country can be done. So actually, what happens? The whole world have accepted that particular standard, that 20 core codis loci. So in Europe also and in the rest of the world, people are believing that. Standard software and computer formats so that data can be transferred between laboratories and a secure computer network to connect the various sites involved in the database. So that is another challenge. You can say another limitations of this particular, because it should not be hackable. It means hackers should not hack this, because if there is a hacking in this database, then there will be disaster. Then finally, the quality standards so that everyone can rely on the results from each laboratory. So laboratory should be accredited. So this is just for your knowledge that how many DNA samples which are stored in different countries of Europe. This is just latest report, not latest, but 2016 report of NSSI. Right. So Germany is having, you can say 82, you can say 820 million, 82 million, then 8 million, 5 million, 60. This is, you can say, the percentage of the population. You can see the UK is having the highest, right, percentage of the population, which is the DNA is stored in the database, right? And that's why the conviction rate in UK is very high because of this only. Why? Because if the sample is already stored in the database, then any person who is committing crimes once again immediately will be caught based on DNA, right? So that is the benefit of the DNA database. In India, still waiting for the DNA bill. Actually, it was published. It was passed in Lok Sabha, but it is pending in Rajya Sabha, right? Still, it is pending, and I hope that we hope that the decision will be taken very soon, and the DNA bill will come in existence, so that all the forensic experts are also getting some jobs, and it will be beneficial for the justice delivery system also, right? Now, what are the contemporary developments? I hope I am having time. Dr. Indu?
Yes, sir. Please go on.
So, okay, I can continue.
Right.
Okay, so yeah, still I want to introduce some modern technology which are there, which can be utilized to further strengthen the power of forensic genetics. You can see this rapid hit technology introduced by Thermo Fisher recently, just 2 years back, and which is, you can say, touch and go system, just plug and play system. Even a small police constable can handle DNA profiling. There is no need to know about this rocket science. Just take the cartridge, collect the sample, put it inside this, this cartridge inside this, just load it and press the button start. That's all. Within 90 minutes, you will get from sample to DNA profile. No need to do the PCR directly. No need to do the DNA extraction. No need to do the gel electrophoresis. Immediately, everything will be done in the single machine, and you will get the profile within 90 minutes. So why this is helping? Why? Because when the person is caught on the site, and if you want to do the DNA analysis, immediately on site you can do the analysis, and you can send the profile to the database. If it is matching with somebody else, or if it is having some evidence, definitely you can link that person with other crimes also, or with some other evidences also. Okay, so this is very much it is, you can say, wonderful instrument, and the Europe and USA has adopted it, and it is having a very good positive result in terms of conviction and in terms of the catching the perpetrators. Another next generation sequencing, NGS. I'll tell you, this particular NGS, the next generation sequencing technology, is, you can say it's a very latest and a very advanced technique, but still the admissibility of this particular technique is on question mark. The reason, the most important reason for this NGS is, by this NGS you can know other traits of persons also. It means you can know that whether the person is going to be suffer with cancer, whether the person is diabetic, whether the person is having particular genetic disease, right? Because it will reveal your privacy. And because of this privacy act, and because of this privacy rules, which are strict in terms of legal aspect, these NGS have not taken place in justice delivery system, but it is utilized nowadays just for lead, right? When you don't have, means when there is an unknown sample with you, right? For example, mass disaster victim identification, in that you can use this, right? When there is unknown dead bodies are there, and you want to identify the you can say the, for example, you are getting only bone, right? Bone sample or a tissue sample, and from that you want to know about the hair color, eye color, and lot many other traits. NGS will give you the answer, right? There are three different technologies which are there. This is the first sequencing technology which was considered to be NGS. Then the second platform came with the sequencing by ligation by Applied Biosystem, and finally Illumina, that is you can say sequencing by bridge amplification technology, right? So all these three technologies are, pyrosequencing is now absolute, but these two technologies are still existing, and they are advancing in much more. And because of this particular technology, the sequencing cost came down, and because of that, people are very hopeful that NGS will get place in the justice delivery system, and it will solve lot many other cases which are next to impossible to solve. From genotyping to forensic phenotyping, it is one of the most fascinating application of next generation sequencing, that you can know the hair color and skin color and eye color based on this single nucleotide polymorphism. I'm not going much more detail, but you can visit HiRiskPlex. If you will type in Google HiRiskPlex, you'll come to know about this particular kit, and you can have the determination of eye color, and you can have a prediction of this particular thing. Microbial forensics, it is still, you can say, budding science in forensic application, which can give a complete, you can say, the very accurate PMI, post-mortem interval. Actually, when your body decays in advanced stage like this particular, D and E, so you cannot tell actually how much time has has passed after the death, but microbes can tell you very accurately up to 180 days, right? So that is very amazing. Right. Then, in case of diagnosis of drowning, right, when you fail with the conventional techniques, microbial techniques will give you idea that okay, you can even connect one particular animal with that particular geographical locations also. Similarly, pathogen detection. So that is already used in particular thing, but whenever there is a bio weapon or identification, at the time this particular microbial forensics can help. Then barcode of live database, bold. So non-human identification, when you want to identify animals, plant species, this is the destination. See, this is one of the biggest database which is containing, you can say, the diversity from lots of countries, and still growing, right? This is the situation of 2015. But nowadays, there are millions of data is available, and you can identify animal and plant based on some markers. Just like human, we are having STRs, but every animal is not having STR. Few animals are characterized for the STR, but most of the animals species can be identified based on the mitochondrial markers that are cytochrome oxidase 1 and cytochrome B. Right. Plant species are also there. There are a number of other markers which can be utilized to identify the plants. Then molecular autopsy, right? So I'm not going much more detail, but this is a really fascinating field in forensic genetics. Basically, it is connected with the pharmacogenomics and pharmacogenetics. So it is one of the examples you can see. This was published in Lancet in 2006. You can see that before almost 15 years back, right? It was published. And how they did it, what was the case? Basically, the 13-day-old baby was died from morphine intoxication, and the reason was unknown in the hospital. And when everything was failed, so the scientists, when they analyzed the woman's, that mother's genome, they came to know that whatever the painkiller which was given to that mother, which was converted to the morphine, right? Because that mother was having multiple copy of that gene, and which was the rare event in that population. And that's why the morphine came to the breast milk, and finally the baby died. Right, and that was, but you can see this is alarming. It sometimes the death is, you can say the cause of death is not known. You don't know how this particular happened. So at that time, this molecular autopsy may work. Because of that, you can say individualization genomics, right? This kind of cases can be solved and can be prevented, right? Similarly, related allergy related, when nowadays doctors asking you many times, so if you are allergic to that particular drug, so it is known for only few drugs. But what if you are allergic to particular drug and it is not known, right? So when you are having such kind of adverse reactions, this particular field can help you. What are the further limitations and challenges in forensic genetics? The CSI and CID fact. This is really one of the most dangerous situations in the society which is prevailing because of these two series. This series is very well known in India, and this series is across the world, which is well known. And because of that, what happened? The you can say utopia-like expectation is arising in the society, right? Because you can say this very interesting gentleman get any analysis to be done. The same person is doing toxicological analysis, biological analysis, physical analysis, and everything. So because of this, then it will give you a false image of the DNA. Everybody is believing that when the DNA is there, the case can be solved. It is not true. It is a myth, right? So DNA is prone to degradation. DNA is a very sensitive technique. That's why it is prone to contamination. The false allegations, false argument in the court will be there. And most of the lawyers, many lawyers, results to even judges also, right? And because of this series, there is a problem. Another one is, which are the neglected questions are there? How was the sample deposited? So sometimes, the sample is deposited by accident. For example, by family member only. So at that time, how to explain that? That is still unclear. When you are connecting it with the DNA, how long will it persist there? For example, after 10 months, you are taking that particular material and still the DNA is there. Then can you tell that that person is done, or that 10 years before that person handled that material has committed that particular crime? How will you prove that? It is still unclear. Will it be transferred further? It means secondary transfer. That is also a wonderful case of the secondary transfer that was in Europe. What happened? That lady, which was packing the swabs, she was unknowingly depositing her DNA on those swabs, and those swabs, when utilized throughout the Europe, a ghost profile of a female was coming in every cases. And everybody was very surprised to know that. And finally, after 3 to 4 years, the case was solved, That was the problem, right? So that is very important because PCR is very sensitive. How can it be detected, recovered, or contaminated? So these are also the sources are also very unclear. These are the definitely challenges that you can say: the temperature when you are storing it, humidity, the bacterial contamination, and of course time. How much time has lapsed after the collection of the sample or the deposition of the sample. So that is degrading the DNA. Finally, last but not the least, the combination knowledge gap between the crime scene investigator and laboratory scientist. Right? They are working in the lab and they are working in the field. They are I'm talking especially in Indian scenario. They are telling that we are knowing everything about the crime scene sample collection, and they are telling that they don't means these people don't know, means they do know nothing about the samples, and it is true also many times because how to handle those particular samples, still the education is not that much clear to most of the constables and most of the police officers, and that's why many a time the crucial samples are destroyed or overlooked by this kind of experts. So there is a knowledge gap between this, right? Whatever the knowledge they are having, they don't want to transfer here, and whatever the knowledge they have, that they are thinking that we are sufficient with the knowledge. So that is a problem. Another example of the DNA forensic genetics failure is Innocence Project. If you have heard about this particular, it is really interesting to visit this particular site. I have taken snap from the website itself just today only. You can see this Joseph Abbott. If you will read the story of this person who served 14 years in the jail, and after 14 years it was proved that he was innocent. Right? You can see here the person, Christopher Abernathy. 28 years he spent in jail, and finally he was released that you are innocent based on DNA. So it is, the Innocence Project is proving both the things, it's both the side of the DNA, right? Sometimes the person was convicted based on the DNA, and now it is proved based on the DNA, once again innocent. And sometimes the person was convicted lack of DNA evidences, but now DNA evidences are there, and that's why he was released, right? So both the sides are involved here. So how to tell whether it is a good or bad? It depends on the case, right? So if you will ask the power of DNA technology, you can ask Mr. Michael Morton. You can see this person, right? Nearly in 2011, that is the case of Innocence Project only. Forensic analysis of DNA samples from a crime scene led to the release of this particular person from prison after he served nearly 25 years for a crime he did not commit, the brutal murder of his wife. You can see the, who will give 25 years back to him? Nobody can give, right? But yes, DNA technology solved this case after 25 years. So he's having still rest of the life to live with, free, right? So this is the, you can say, power of DNA technology. But at the same time, sometimes, there is a contamination, there is a mixture profile. So at that time, it will be very difficult to analyze this kind of samples, and it will pose a very big challenge for that. And finally, the most important challenge in forensic genetics is this CRISPR-Cas technology. Everybody is knowing about this word. I don't know how many of you have gone in much more detail how you can edit it, but you must be knowing this person. If you are not knowing, go to the website and search about the designer baby controversy in China. You will definitely get this picture. It is Dr. He, right? And he created 2 babies with CRISPR-Cas technology, and which was banned, which is still banned. And he was sentenced 3 years imprisonment. And finally, what he did is basically he actually did good for these babies. Actually, he cured these babies from one metabolic diseases, right? But the trials are not allowed on this particular thing using this CRISPR-Cas technology for human. Still, it is not allowed in China. So that's why the controversy was there. And designer babies. So just imagine if one scientist will erase all the STR markers from one person, and that person becomes a big criminal in future, what will happen? Will you able to catch it with the DNA samples? Not at all, because all the markers are erased. Simple, because all the markers are in non-coding DNA. and these particular markers are not means it's not required for further function of the DNA. Till today, it is believed to be like that. We don't know the function of that particular. But if you knock out those particular markers, what will happen? You'll be a ghost, right? It means a ghost criminal. You cannot be catched by DNA at all, right? So this is the biggest problem. Another problem is that because of this CRISPR and you can say artificial reproductive techniques, lots of complications are occurring between different paternal issues, right? I think if you have heard about the baby with a three parent, right? Two mothers and one father, right? Because of the one mother donated the egg, another mother donated the nucleus, and the father donated the sperm. So this is how this baby. Why? Because the baby was suffering from the mitochondrial disease, right? Mitochondrial DNA based disease, and that's why the mitochondria were taken from the healthy mother, and the nucleus was taken from that original mother, and the sperm was donated by the father. So this is how the baby became, having three parents. So that is also becoming a social issues nowadays, right? Surrogacy issues are there, and lots of issues are there because of the sperm donations and artificial reproductive treatment or techniques. So let's see how we can go further and solve these problems. That's all. These are the references, few references that I've taken, and few articles also I have searched for this, which are not shown here, right? Then thank you for being a wonderful audience. So that's all for the day.
Thank you, sir. Thank you so much. Sir, you have truly explained this very complicated topic with such ease, in such lucid language, and I'm sure that everyone assimilated a lot from that. So if you will, can we move on to the question answer session? Because
ICMR approved. We are not doing public testing, but it is we are doing some it is for some research purpose.
So the next question, is there any time limit for taking fingerprint for examination?
Okay, time limit. See, fingerprint. So they are talking about the fingerprint. So fingerprint. See, is he or she asking about latent fingerprint or DNA fingerprint?
They have not specified, but I assume it must be DNA fingerprinting because that is related to the topic. So if you could explain about that.
Okay. So if it is for DNA fingerprint, then as soon as possible, right? That is the rule. But there is no time limit for any samples to be collected, because many a times, the sample is 1 year old, 2 year old. Sometimes it is giving profile also if it is well preserved. It depends on how environmental condition is prevailing around that particular sample. And if he is talking or she is talking about the latent fingerprint, then latent fingerprint is having some time limit, right? So sometimes, within 2 or 3 days, it fade away. But DNA, because it is open, sometimes it is open. So bacterial contamination will be there. And if the water activity is there, then it will be definitely degraded very soon. But you should try to wipe it out, and you should try to do the DNA analysis at least.
So we have another question from Shreya. Can anyone be born without fingerprint due to genetic aberrations?
Yeah, I have heard about the cases that fingerprint is not there, right? I have heard about that cases. But for your information, fingerprint has nothing to do with the DNA, because fingerprints are developed outside of the, because of the womb activity and some other physiological features, right? So there is no genetic relationship between the fingerprint and, because both are unique and both are highly different.
So next question is from Srirag. If a situation occurs that we get only one drop of blood from the crime scene and the suspects we have are identical twins.
Okay, so when in case of identical twins with current STR technology, you cannot differentiate. But if you are using a DNA methylation technology, it is possible to differentiate identical twins with the DNA, because the DNA expression pattern will get changed in both the identical twins. So based on that DNA profiling, RNA profiling, you can know the difference between this identical twins. The technology is there.
So another question, which is more important for investigation, nuclear or mitochondrial DNA?
Obviously nuclear, because mitochondrial DNA will give you the only maternal lineage. It will not give you the identification of the person. It will not individualize the person. It will give you the maternal lineage. You can link with the family members, but you cannot identify a person. But yeah, nuclear is there. Definitely you can individualize the person.
And I think this is the last question we'll be taking for this session. In which gestational time period does fingerprint start to form? I think, sir.
See, I'm not much more clear about that because I have not gone much more developmental biology in detail. So, in which gestation period, I don't know about that exactly. Right.
So I think that's all for questions in the session. So there's one more question. Please tell us about the BISRA report in brief.
BISRA report or VISRA?
Tarun sir, if you could maybe clarify your question a bit. In the meantime, sir, thank you so much for spending so much time. VISRA.
VISRA.
Yes.
VISRA is a completely toxicological analysis, right? When some any toxicological analysis is to be done, at the time the VISRA is sent for the toxicological analysis. So that is, I think, it is completely a chemical aspect, right? So what kind of drugs, what kind of medication symptoms, for that basically the VISRA is sent, right? So it is not for the DNA fingerprinting.
Right. Thank you so much, sir. Thank you for spending so much time, and you've actually taken up a topic which is a nightmare to a lot of students, very difficult to understand, and you've made it very easy, very lucid. And I'm very sure that the lack of questions here show the fact that everyone has assimilated everything. Thank you once again, sir. Thank you so much.
I'm also glad that I have, once again, it basically, by these way, I'm learning only, right? So it makes my fundamental much more clear about that. And there are really good questions, and because of that, even I have to go in much more detail. So I'll go definitely for gestation period and all that. So that is my homework. Okay. So thank you very much for asking for this question.
Thank you, sir. Before you leave, I'd like to present you with a certificate of appreciation for your valuable time and all the knowledge that you shared with all of us. Janita, if you could please.
Thank you very much. Should I stop my sharing?
Yes, sir. Yes, sir. Please.
Yeah,
so, can you see? I hope you can see the certificate.
Just a minute. Let me unpin me. Yeah. Thank you very much.
Thank you so much, sir. Thank you. Now everything's virtual. This should be handed over to you, but again, it's all.
No. In virtual world, it is perfectly okay, and that basically this certificate means a lot to me because Dr. Shiva Sankaran's signature is there, right? And he is really good senior to me. We did Ph.D. in same university, and he was senior to me, and I'm really happy to see his name here. Thank you very much for this.
Thank you, sir. Please stay connected with us.
Yeah, thank you.
Thank you.
So should I leave?
Yes, sir. Yes, sir. Thank you so much.
Yeah, thank you.
Thank you to all the participants too for joining and for staying with us. We've gone half an hour past the decided time. Thank you. The feedback link will be sent via email to all the registered participants along with the certificates. Thank you so much. You may all exit the group. We'll see you next week. Thank you.